plasma membrane or cell membranes are crucial to the life of the cell. The plasma membrane encloses the cell, defines its boundaries, and maintains the essential differences between the cytosol and the extracellular environment. Inside eukaryotic cells, the membranes of the endoplasmic reticulum, Golgi apparatus, mitochondria, and other membrane-enclosed organelles maintain the characteristic differences between the contents of each organelle and the cytosol. Ion gradients across membranes established by the activities of specialized membrane proteins can be used to synthesize ATP to drive the transmembrane movement of selected solutes, or in nerve and muscle cells to produce and transmit electrical signals. In all cells, the plasma membranes also contains proteins that act as sensors of external signals, allowing the cell to change its behavior in response to environmental cues. These protein sensors or receptors transfer information rather than ions or molecules across the membrane. Despite their differing functions, all biological membranes have a common general structure. Each is a very thin film of lipid and protein molecules held together mainly by non-covalent interactions. Cell membranes are dynamic, fluid structures, and most of their molecules are able to move about in the plane of the membrane. The lipid molecules are arranged as a continuous double layer about 5 nanometer thick. This lipid bilayer provides the basic fluid structure of the membrane and serves as a relatively impermeable barrier to the passage of most water-soluble molecules. Protein molecules that span the lipid bilayer mediate nearly all of the other functions of the membrane, transporting specific molecules across it, for example, or catalyzing membrane-associated reactions such as ATP synthesis. In the plasma membrane, some proteins serve as structural links that connect the cytoskeleton through the lipid bilayer to either the extracellular matrix or an adjacent cell, while others serve as receptors to detect and transduce chemical signals in the cell's environment. As would be expected, it takes many different membrane proteins to enable a cell to function and interact with its environment. In fact, it is estimated that about 30% of the proteins that are encoded in an animal cell's genome are membrane proteins. Various hypotheses or models of plasma membrane. A convenient starting point about the models of membrane structure is the pioneering work of the scientist Charles Ernst Overton in the 1890s. In the late 19th century, he investigated the osmotic properties of cells and noticed that the permeation of molecules through membranes is related to their partition coefficient between water and oil. From his studies, Overton led to the hypothesis that the thin membranes surrounding cell have a properties of oil and are lipids which regulate the permeability of the cell. Therefore, working with cells of plant root hairs, he observed that lipid-soluble substances penetrate readily into cells, whereas water-soluble substances do not. Langmuir in 1917. A decade later, a second significant advance came through the work of Irving Langmuir. He studied the behavior of purified phospholipids by dissolving them in benzene and layering samples of the benzene lipid solution onto a water surface. As the benzene evaporated, the molecules were left as a lipid monolayer. Since phospholipids are amphipathic molecules, Langmuir concluded that the phospholipids orient themselves on water such that their hydrophilic heads face the water and their hydrophobic tails protrude away from the water. Therefore, Langmuir's lipid monolayer became the underlying support or foundation in the studies of the membrane structure. Gorter and Grendel in 1925 revealed that cell membranes are made of two opposing thin molecular layers, and they proposed that this double layer is constructed such that two lipid layers form a bilayer with the polar head groups pointing towards the aqueous environment. Danieli and Davison in 1935, they proposed that proteins are adsorbed to the lipophilic layers surrounding cells. The proteins possesses hydrophobic interiors and a water-containing outer layer. The lipid layer possesses amphiphilic or charged head groups. This implies that the lipid membranes also contains some water. The water-containing regions of protein layers adsorbed on lipid layers are permeable for charged solutes, for example, ions. Divalent cations as calcium form complexes with lipids or proteins that reduce their interaction with water. Therefore, membranes containing calcium are less permeable for ions. Hydrophobic molecules such as ether penetrate the membranes through their lipophilic lipid parts. They use their model to interpret the observation of different membrane permeability of ions and hydrocarbons. In particular, they assume that the membrane has both a lipophilic and a hydrophilic character. Robertson in 1958, Robertson pursued his interest in understanding membrane architecture in a series of outstanding early ultrastructural papers, which led soon to his famous unit membrane hypothesis. This he first articulated at an American Physiological Society meeting in 1957. His hypothesis held that all cell membranes, those that form intracellular organelles as well as plasma membranes, are composed of bimolecular leaflets of phospholipid with monolayers of proteins adsorbed to their surfaces. To support this theory, Robertson relied heavily on a particular type of tissue preparation for electron microscopy, which invariably made all biological membranes look like pairs of dark lines in thin sections. This double membrane image he interpreted as resulting from staining of protein monolayers on either side of an unstained central core comprising the bimolecular leaflet of lipids. The fluid mosaic model of Singer and Nicholson, 1972. In the 1960s, the structure of a number of soluble proteins were solved by X-ray crystallography. Leonard and Singer in 1966 found that many membrane proteins have a high alpha helical content. Also, electron micrographs revealed that labeled proteins form isolated spots in some membranes. Furthermore, they considered the role of hydrophobic amino acids in alpha helices. Hence, membranes are constructed from lipids and proteins. From this, Singer and Nicholson concluded that proteins may also span through membranes. This led to the famous Singer-Nicholson model, also known as the fluid mosaic model. Singer and Nicholson in 1972 underlined that some proteins seem to interact with the surrounding lipids, and that protein function may depend on the presence of specific lipids. They propose that the proteins are surrounded by a layer of strongly interacting lipids, while most of the remaining lipids are hardly influenced by the presence of proteins. This implies that the lipids form a matrix and no long range order of protein exists within the matrix. Short range order due to protein-protein interactions, possibly mediated by specific lipids, was considered as possibility. It was postulated that the lipid membranes of biological cells are in the fluid lipid state, with exceptions, for example, the myelin, in which proteins can freely diffuse. Singer and Nicholson also noted that the fluid mosaic membrane is most likely asymmetric and displays distinctly different features on the inside and outside of cells. The term fluid mosaic model used by Singer and Nicholson probably originates from Danieli and Dawson in 1935. They basically confirmed the models of Gorter and Grandel 1925 and Danieli and Dawson 1935, basing on microscopic evidence. Robertson's model was sometimes incorrectly interpreted as that all membranes have the same composition. However, Robertson's statement was merely meant to describe a common structure. The Singer-Nicholson model still is the widely accepted model. In particular, due to progresses in the crystallization of membrane proteins, it is nowadays known that membrane proteins displays alpha helical or beta barrel-like membrane spanning segments of predominantly polar amino acids. In the fluid mosaic model, which has served as the central dogma of membrane biology for 3 decades, the lipid bilayer remains the core of the membrane, but the attention is focused on the physical state of the lipid. Unlike previous models, the bilayer of a fluid mosaic membrane is present in a fluid state, and individual lipid molecules can move laterally within the plan of a membrane. The structure and arrangement of membrane proteins in the fluid mosaic model differ from that of previous models in that they occur as a mosaic of discontinuous particles that penetrate the lipid sheet. Most importantly, the fluid mosaic model presents cellular membranes as dynamic structures in which the components are mobile and capable of coming together to engage in various types of transient or semi-permanent interactions. In the following sections, we will examine some of the evidence used to formulate and support this dynamic portrait of membrane structure and look at some of the recent data that bring the model up to date. A fluid mosaic model is presented for the gross organization and structure of the proteins and lipids of biological membranes. The model is consistent with the restriction imposed by thermodynamics. In this model, the proteins that are integral to the membrane are a heterogeneous set of globular molecules, each arranged in an amphipathic structure, that is, with the ionic and highly polar groups protruding from the membrane into the aqueous phase and the nonpolar groups largely buried in the hydrophobic interior of the membrane. These globular molecules are partially embedded in a matrix of phospholipid. The bulk of the phospholipid is organized as a discontinuous fluid bilayer, although a small fraction of the lipid may interact specifically with the membrane proteins. The fluid mosaic structure is therefore formally analogous to a two-dimensional oriented solution of integral proteins or lipoproteins in the viscous phospholipid bilayer solvent. Recent experiments with a wide variety of techniques and several different membrane systems are described, all of which a bit consistent with and add much detail to the fluid mosaic model. It therefore seems appropriate to suggest possible mechanisms for various membrane functions and membrane-mediated phenomena in the light of the model. As examples, experimentally testable mechanisms are suggested for cell surface changes in malignant transformation and for cooperative effects exhibited in the interactions of membranes with some specific ligands. The matrix model by Moritzen and Bloom in 1984. The fluid mosaic model of Singer-Nicolson has nowadays experienced some refinement, which takes into account that lipids and proteins may distribute inhomogeneously and that domains and clusters may form within the membrane. Without explicitly saying so, the Singer-Nicolson model considered the lipid membranes as a homogeneous fluid in which the proteins diffuse in two dimensions. In 1984, Moritzen and Bloom proposed the matrix model that suggests that proteins and lipids display interactions with a positive free energy content due to variations in the hydrophobic length of the molecules. The typical thickness of a lipid bilayer is about 5 nanometer. If the hydrophobic core of a membrane protein is longer or shorter than this length, either some hydrophobic proteins or lipid segments are exposed to water, or the lipid membranes has to be deformed to compensate for the unfavorable hydrophobic interactions. This effect is called the hydrophobic matching. The hydrophobic matching gives rise to interfacial tensions between lipids and proteins. These tensions may result in the accumulation of certain lipid species around the proteins and in the mutual attraction of proteins due to capillary forces, leading to aggregation and clustering of proteins. Let us talk about a general structure of plasma membrane. Plasma membranes are lipid protein assemblies in which the components are held together in a thin sheet by non-covalent bonds. As noted from the above models, the core of the membrane consists of a sheet of lipids arranged in a bimolecular layer. The lipid bilayer serves primarily as a structural backbone of the membrane and provides the barrier that prevents random movements of water-soluble materials into and out of the cell. Each type of differentiated cell contains a unique complement of membrane proteins, which contributes to the specialized activities of that cell type. The ratio of lipid to protein in a membrane varies depending on the type of cellular membrane, such as plasma versus endoplasmic reticulum versus Golgi, the type of organism such as bacterium versus plant versus animal, and the type of cell such as cartilage versus muscles versus liver. For example, the inner mitochondrial membranes has a very high ratio of protein lipid in comparison to the red blood cell plasma membrane, which is high in comparison to the membranes of the myelin sheath that form a multi-layered wrapping around a nerve cell. To a large degree, these differences can be correlated with the basic functions of these membranes. The inner mitochondrial membrane contains the protein carriers of the electron transport chain, and relative other membranes, lipid is diminished. In contrast, the myelin sheath acts primarily as electrical insulation for the nerve cell it encloses, a function that is best carried out by a thick lipid layer of a high electrical resistance with a minimal content of protein. Membranes also contain carbohydrates, which are attached to the lipids and proteins. Membrane lipids. Biological membranes consist of a continuous double layer of lipid molecules in which membrane proteins are embedded. This lipid bilayer is fluid with individual lipid molecules able to diffuse rapidly within their own monolayer. The membrane lipids molecules are amphipathic. The most numerous are the phospholipids. When placed in a water, they assemble spontaneously into bilayers, which form sealed compartments that reseal if torn. The lipid compositions of the inner and outer monolayers are different, reflecting the different functions of the two faces of a cell membrane. Different mixtures of lipids are found in the membranes of cells of different types, as well as in the various membranes of a single eukaryotic cell. Some membrane-bound enzymes require specific lipid head groups in order to function. The head groups of some lipids form docking sites for specific cytosolic proteins. Some extracellular signals that act through membrane receptor proteins activate phospholipases that cleave selected phospholipid molecules in the plasma membrane, thereby generating fragments that act as intracellular signaling molecules. Membranes contains a wide diversity of lipids, all of which are amphipathic, that is, they contain both hydrophilic and hydrophobic regions. There are three main types of membrane lipids: phosphoglycerides, sphingolipids, and cholesterol. Phosphoglycerides. Most membrane lipids contain a phosphate group, which makes them phospholipids. Because most membrane phospholipids are built on a glycerol backbone, they are called phosphoglycerides. Unlike triglycerides, which have 3 fatty acids and are not amphipathic, membrane glycerides are diglycerides. Only 2 of the hydroxyl groups of the glycerol are esterified to fatty acids. The third is esterified to a hydrophilic phosphate group. Membrane phosphoglycerides have an additional group linked to the phosphate, most commonly either choline, ethanolamine, serine, or inositol. Each of these groups is small and hydrophilic, and together with the negatively charged phosphate to which it is attached, forms a highly water-soluble domain at one end of the molecule called the head group. At physiologic pH, the head groups have an overall negative charge. A membrane fatty acid may be fully saturated, that is, lack double bonds. Mono unsaturated, that is, possesses one double bond, or polyunsaturated, that is, possesses more than one double bond. Phosphoglycerides often contain one unsaturated and one saturated fatty acyl chain. Sphingolipids. A less abundant class of membrane lipids called sphingolipids are derivatives of sphingosine, an amino alcohol that contains a long hydrocarbon chain. Sphingolipids consist of sphingosine linked to a fatty acid by its amino group. The various sphingosine-based lipids have additional groups esterified to the terminal alcohol of the sphingosine moiety. If the substitution is phosphorylcholine, the molecule is sphingomyelin, which is the only phospholipid of the membrane that is not built with a glycerol backbone. If the substitution is a carbohydrate, the molecule is a glycolipid. If the carbohydrate is a simple sugar, the glycolipid is called a cerebroside. If it is a small cluster of sugars, the glycolipid is called a ganglioside. Since all sphingolipids have two long hydrophobic hydrocarbon chains at one end and a hydrophilic regions at the other, they are also amphipathic and basically similar in overall structure to the phosphoglycerides. Glycolipids are interesting membrane components. Relatively little is known about them, yet tantalizing hints have emerged to suggest they play a crucial roles in cell function. The nervous system is particularly rich in glycolipids. The myelin sheath contains a high content of a particular glycolipid called galactocerebroside. Humans who are unable to synthesize a particular ganglioside suffer from a serious neurological disease characterized by severe seizures and blindness. Glycolipids also play a role in certain infectious diseases. The toxins that cause cholera and botulism both enter their target cell by first binding to cell surface gangliosides, as does the influenza virus. Cholesterol. Another lipid component of certain membranes is the sterol cholesterol, which in certain animal cells may constitute up to 50% of the lipid molecules in the plasma membrane. Cholesterol is absent from the plasma membranes of most plant and bacterial cells. Cholesterol molecules are oriented with their small hydrophilic hydroxyl group towards the membrane surface and the remainder of the molecule embedded in the lipid bilayer. The hydrophobic ring of a cholesterol molecule are flat and rigid, and they interfere with the movements of the fatty acid tails of the phospholipids. Importance of the lipid bilayer. The role of this remarkable diversity of lipid species remains the subject of interest and speculation. The lipids of a membrane are more than simple structural elements. They can have important effects on the biological properties of a membrane. Lipid composition can determine the physical state of the membrane and influence the activity of particular membrane proteins. Membrane lipids also provide the precursors for highly active chemical messengers that regulate cellular function. Because of thermodynamic considerations, the hydrocarbon chains of the lipid bilayer are never exposed to the surrounding aqueous solution. Consequently, membranes are never seen to have a free edge. They are always continuous, unbroken structures. As a result, membranes form extensive interconnected networks within the cell. Because of the flexibility of the lipid bilayer, membranes are deformable and their overall shape can change as occurs during locomotion or cell division. The lipid bilayer is thought to facilitate the regulated fusion or budding of membranes. The importance of the lipid bilayer in maintaining the proper internal composition of a cell in separating electric charges across the plasma membrane and in many other cellular activities will be apparent throughout this and subsequent chapters. Another important features of the lipid bilayer is its ability to self-assemble, which can be demonstrated more easily within a test tube than a living cell. If, for example, a small amount of phosphatidylcholine is dispersed in an aqueous solution, the phospholipid molecules assemble spontaneously to form the walls of a fluid-filled spherical vesicles called liposomes. The walls of these liposomes consist of a continuous lipid bilayer that is organized in the same manner as that of the lipid bilayer of a natural membrane. Membrane carbohydrates. The plasma membranes of a eukaryotic cells also contain carbohydrate. Depending on the species and cell type, the carbohydrate content of the plasma membrane ranges between 2 and 10% by weight. More than 90% of the membrane's carbohydrate is covalently linked to proteins to form a glycoproteins. The remaining carbohydrate is covalently linked to lipids to form glycolipids. Carbohydrate of the plasma membrane faces outward into the extracellular space. The carbohydrate of glycoproteins is present as short, branched hydrophilic oligosaccharides, typically having fewer than about 15 sugars per chain. In contrast to most high molecular weight carbohydrates such as glycogen, starch or cellulose, which are polymers of a single sugar, the oligosaccharides attached to membrane proteins and lipid can display considerable variability in composition and structure. Oligosaccharides may be attached to several different amino acids by two major types of linkages. Importance. The carbohydrates of the glycolipids of the red blood cell plasma membrane determine whether a person's blood type is A, B, AB or O. A person having blood type A has an enzyme that adds an N-acetyl-galactosamine to the end of the chain, whereas a person with type B blood has an enzyme that adds galactose to the chain terminus. These two enzymes are encoded by alternate versions of the same gene, yet they recognize different substrates. People with AB blood type possesses both enzymes, whereas people with O blood type lack enzymes capable of attaching either terminal sugar. Membrane proteins. Whereas the lipid bilayer determines the basic structure of a biological membranes, proteins are responsible for most membrane functions, serving as specific receptors, enzymes, transport proteins and so on. Many membrane proteins extend across the lipid bilayer. In some of these transmembrane proteins, the polypeptide chain crosses the bilayer as a single helix, that is called single pass protein. In others, including those responsible for transmembrane transport of ions and other small water soluble molecules, the polypeptide chain crosses the bilayer multiple times, either as a series of alpha helices or as a beta sheet in the form of a closed barrel, that is called multi-pass proteins. Other membrane associated proteins do not span the bilayer, but instead are attached to either side of the membrane. Many of these are bound by non-covalent interactions with transmembrane proteins, but others are bound via covalently attached lipid groups. Like the lipid molecules in the bilayer, many membrane proteins are able to diffuse rapidly in the plan of the membrane. However, cells have always of immobilizing specific membrane proteins and of confining both membrane proteins and lipid molecules to particular domains in a continuous lipid bilayer. In the plasma membrane of all eukaryotic cells, most of the proteins exposed on the cell surface and some of the lipid molecules in the outer lipid monolayer have oligosaccharide chains covalently attached to them. Plasma membranes also contain integral proteoglycan molecules with surface exposed polysaccharide chains. The resulting sugar coating is though to protect the cell surface from mechanical and chemical damage. In addition, some of the oligosaccharide chains are recognized by cell surface carbohydrate binding proteins, for example lectins, that help mediate cell-cell adhesion events. Depending on the cell type and the particular organelles within that cell, a membrane may contain hundreds of different proteins. Each membrane protein has a defined orientation relative to the cytoplasm, so that the properties of one surface of a membrane are very different from those of the other surface. This asymmetry is referred to as membrane sidedness. In the plasma membrane, for example, those parts of membrane proteins that interact with other cells or with extracellular substances project outward into the extracellular space, whereas those parts of the membrane proteins that interact with cytoplasmic molecules project into the cytosol. Membrane proteins can be grouped into three distinct classes distinguished by the intimacy of their relationship to the lipid bilayer. First is integral proteins that penetrate the lipid bilayer. Integral proteins are transmembrane proteins, that is, they pass entirely through the lipid bilayer and thus have domains that protrude from both the extracellular and cytoplasmic sides of the membrane. Some integral proteins have only one membrane spanning segment, whereas others are multispanning. Peripheral proteins that are located entirely outside of the lipid bilayer on either the cytoplasmic or extracellular side, yet are associated with the surface of the membrane by non-covalent bonds. Lipid anchored proteins that are located outside the lipid bilayer on either the extracellular or cytoplasmic surface, but are covalently linked to a lipid molecule that is situated within the bilayer. Now let us see importance of membrane fluidity. Membrane fluidity provides a perfect compromise between a rigid, ordered structure in which mobility would be absent and a completely fluid, non-viscous liquid in which the components of the membrane could not be oriented and structural organization and mechanical support would be lacking. In addition, fluidity allows for interactions to take place within the membrane. For example, membrane fluidity makes it possible for clusters of membrane proteins to assemble at particular sites within the membrane and form specialized structures such as intercellular junctions, light capturing photosynthetic complexes and synapses. Because of membrane fluidity, molecules that interact can come together, carry out the necessary reaction and move apart. Membrane arise only from pre-existing membranes, and their growth is accomplished by the insertion of lipids and proteins into the fluid matrix of the membranous sheet. Many of the most basic cellular processes, including cell movement, cell growth, cell division, formation of intercellular junctions, secretion and endocytosis, depends on the movement of membrane components and would probably not be possible if membranes were rigid, non-fluid structures. In concluding the topic, It can be said that cell membranes are important to the life of the cell. It encloses the cell, defines its boundaries, and maintain the essential differences between the cytosol and the extracellular environment. Cell membranes are very dynamic, fluid structures, and most of their molecules are able to move about in the plane of the membrane. Various scientists proposed a different model explaining structures of plasma membrane, but fluid mosaic model of Singer and Nicholson was widely accepted. The fluid mosaic model is dynamic structures in which the components are free to move. Membrane composed of mainly lipid, protein and carbohydrates, and perform various important activities for the cell. Thanks.